Good afternoon. I'm Judy Lieberman, and it's my great pleasure to introduce Marcella Moss for the seminar today. Marcella is an MD PhD who was trained at UPenn. She had the great fortune or smart decision to work in like the premier immunotherapy and gene therapy labs in the country. She got her PhD with Carl June, working on the design of CAR T cells. She had a landmark Nature Biotech paper on a very effective design for CAR T cells. And then she did a short postdoc with Kathy High, looking at the immune response to her pioneering gene therapy, and then went to Cornell and participated in their immunotherapy efforts. So Marcella has been preoccupied with developing and extending and improving CAR T cell therapy. She's been involved in trying to figure out not only how to design CAR T cells so that they work and live long and are functional, but also trying to understand the molecular basis of some of the toxicities and trying to extend CAR T cells from their incredibly effectiveness for B-cell malignancies to solid tumors and also to multiple myeloma. She came to Massachusetts General Hospital a little over 5 years ago, where she's in charge of the immunotherapy efforts in the Cancer Center. And she's a colleague. She's been promoted up the ranks. She's now a chaired associate professor at MGH, and she's a wonderful colleague to have at Harvard in the immunology program. Marcella is going to talk to us today about designing and translating next generation CAR T cells. At the end of her talk, we will take questions, and please put your questions on the Q&A, and I will ask them of Marcella at the end of her talk. Marcella, take it away.
Thank you so much, Judy. Thank you so much for the kind introduction, and also to David for starting this series with our colleagues at DFHCC, of which I'm also fortunate to be part of. So I'm going to talk to you today about designing and translating next generation CAR T cells, and sort of walk you through some of our initial work going back now more than 5 years in glioblastoma, and also tell you about what we've been working on at MGH in the last 5 years or so. These are my disclosures. So for those of you who need a little bit of background in immunotherapy and in T cell therapy in particular, I've been interested in getting T cells to respond to tumor and to kill tumor going back more than 20 years now. And so there's been a variety of ways where this has been attempted. So the first generation methods really involved trying to wake up an endogenous immune response, thinking that T cells had natural T cell receptors that would recognize neoantigens or mutated antigens in tumor cells, that fundamentally tumor was different from self and that it could be recognized by the endogenous T cell repertoire. And initial efforts really revolved around therapeutic vaccines, taking up tumor, lysing it up, getting either RNA or protein or tumor lysate and injecting it and trying to stimulate a T cell response in the way of a vaccine. Another hypothesis was that T cells required additional cytokines to be able to activate, grow, and proliferate and sustain an anti-tumor response. And so there were multiple cytokines that were attempted as primary cytokine therapy to try to wake up endogenous immune responses. Well, this actually, neither one of these systems worked particularly well, and that's partly because there are multiple checkpoints expressed by tumors and in the tumor microenvironment. And so even though there are some pre-existing T cell receptors that could potentially recognize tumors, they were blocked in the tumor microenvironment and even at the level of antigen presentation in the lymph node. And so, of course, the field took a massive leap forward with the advent of first anti-CTLA-4 and then anti-PD-1 and anti-PD-L1 checkpoint blockade. But these are still about waking up endogenous T-cell responses to what could be perceived as native, as foreign antigens that are presented by tumors. These would still all be HLA or MHC restricted. And so kind of in parallel with the development of checkpoint blockade, there were efforts to either clone T cells or engineer T cells with novel immune receptors to be directed to recognize specific tumor antigens that were defined by the investigators. So for example, there were a number of T cell receptors that were cloned and isolated, specifically to cancer testis antigens, for example, that could then be introduced using gene engineering methods, whether that's transposons or retroviral or lentiviral vectors, so that we now had dual specific T cell receptor engineered T cells that would recognize MHC plus peptide combinations of a defined specificity in the tumor. We took a different approach also, and this was initiated by several investigators in the early 90s, using chimeric antigen receptors, where the antigen recognition portions of an antibody were fused to T cell signaling domains. And these had various names, including chimeric immune receptors and T bodies. And then they were named CAR T cells, and it sort of took off as that name in the late 1990s, early 2000s. And so the advantages of CARs is that they are, sorry, having trouble advancing. The advantages of CARs is that they recognize surface proteins in a non-MHC restricted fashion, and that co-stimulation can be engineered into the antigen receptor itself to endow the T cell with co-stimulation and the ability to proliferate or make additional cytokine or resist activation-induced cell death upon engagement of that receptor. And so these CAR T cells have potential applications in multiple tumors as long as there is a tumor-specific surface protein. Now, the disadvantage is that there are not very many tumor-associated antigens that are very tumor-specific and that are expressed on the surface. Most of the sort of cancer-specific repertoire are intracellular antigens, and so these would still need targeting by a TCR. But I'm going to talk to you mostly today about chimeric antigen receptor T cells because these have had tremendous impact, particularly in hematologic malignancies, and I think they still have a lot of potential in solid tumors, which I'll get into as well. So let's talk a little bit about CAR T cell anatomy. You can divvy up a CAR basically by each amino acid, but to spare you that level of detail, we're going to talk about the main portions, which are the antigen binding portion. And these are typically based on single chain variable fragments derived from antibodies, but they can also be based on single domain antibodies or even natural ligands. And I'll show you some data we have with that. They can also be engineered to have different affinities. There's a hinge domain, which separates the antigen binding from the transmembrane domain. And this at first was thought to be biologically inert, but emerging data have suggested that depending on which one you choose, it can actually have biologic effects as well. And having the length of the hinge domain can also impact how the CAR binds its target epitope on the target cell. There is co-stimulation that is typically engineered into CARs. The first generation CARs were ones that did not have co-stimulation. We don't talk about them as much anymore because they didn't have much clinical impact. And part of that was due to a lack of persistence or expansion of the T cells. There wasn't much anti-tumor effect. But once these CARs had embedded co-stimulation, whether that was derived from CD28 or 4-1BB, the story really changed with rapid and persistent expansion of these CARs and strong anti-tumor effects. Most CARs also have the primary T cell activation domain derived from the TCR, which is called CD3. This is the zeta portion of the CD3 complex. This has 3 ITAMs, which generate the first activation signal into the T cell. And there are a variety, of course, of mutations and alternatives that can be generated to design a CAR, but these are the basic, this is the basic anatomy of the CAR. CARs themselves can be placed into different immune effector cells. So my focus is on T cells, but CARs have been engineered into subsets of T cells, including gamma delta cells and Treg cells, and also into NK cells, whether they are cord blood derived or IPS derived. And they can also be engineered into sort of off-the-shelf T cells or off-the-shelf NK cells when they are used in conjunction with gene editing technologies to remove endogenous T cell receptors or to make these sort of stealth so that there's not either graft versus host disease or rejection of the incoming CAR. On the gene therapy side, there's also a variety of ways that CARs can be introduced genetically into the chromosome or the genetic architecture of the target cell. Typically, we use integrating technologies such as retroviral or lentiviral vectors, but you can also use transposons or others, and the CAR T cell can also be shuttled in with additional functions such as PD-1 knockout, or the T cell itself can be additionally modified either with small molecule drugs or even with nanoparticles. So there's an infinite amount of complexity that can be added to what is generically called a CAR T cell. But I'm going to start with this sort of what's called a second generation CAR T and focus on this anatomy first before we go into more complex CAR design. So the first CARs that really had anti-tumor effect in humans were directed towards the human CD19 molecule. Human CD19 is quite special because it's expressed throughout B cell development, and yet the B cell is not an essential cell type for life-sustaining. It's not a life-sustaining cell type, and so it's not expressed on hematopoietic stem cells or anywhere else in the body. And there are multiple tumors that are derived from B cells in various stages of development. And so this allows basically one CAR construct to be developed in clinical trials for multiple indications, such as ALL, mantle cell lymphoma, follicular lymphoma, diffuse large B-cell lymphoma, et cetera. CD19 expression tends to wane in the plasma cell. And so although there is a little bit of data with CD19 CARs for multiple myeloma, it's not as straightforward as the straight up B-cell malignancies. There are now 5 FDA-approved CAR T-cell products. Actually, one has recently achieved a second approval in a different clinical indication, but these are the molecular, this is the molecular anatomy of each of the CARs that is currently clinically approved. Axicell uses a CD28 signaling domain, whereas Tisacell uses a 4-1BB signaling domain. And you'll see that there is a theme here in that most of the CARs have the same anti-CD19 binding region called FMC63, which was the name of the original murine monoclonal antibody to human CD19. There's a couple of different transmembrane domains that are frequently in use, and then most of them use co-stimulation derived from either CD28 or 4-1BB. The last one here on the right is idacaptogene beclucil, or Abecma. This uses a similar construct as what tisagenlecleucel is, but the single chain variable fragment is directed towards human BCMA, B-cell maturation antigen, and this is FDA approved in relapsed or refractory multiple myeloma. In terms of the process to generate CAR T cells, it's quite different than standard monoclonal antibody therapy, chemotherapy, or small molecule tyrosine kinase inhibitor kind of therapy. In most cases, and all of the FDA-approved products are derived from autologous T cells. So patients go to their point of care, get a leukapheresis procedure, have their cells shipped to a central manufacturing facility where they are then activated to enter the cell cycle, genetically modified and transduced, and then they are cultured over a period of 7 to 14 days, and then formulated and frozen and shipped back to the point of care of the patient where they can be infused intravenously. Typically, patients are also giving a preparative course of chemotherapy to enhance homeostatic proliferation of the incoming CAR T cells and improve their expansion. The first evidence that CAR T cells are living drugs came in 2011 when the first 3 adults and the first pediatric patients were treated. And what was really notable is that even though these patients got a single infusion or a set of 3 infusions of CAR T, although they only got around 1 million to 10 million to 100 million cells, the T cells underwent a massive expansion after they were infused into the patient, more than 100,000-fold. And all of these patients had persistence of their CAR T cells in the circulation for the first 6 months when it was reported. And so what we were really seeing is sort of natural T cell biology in terms of an initial exposure to antigen massive proliferation, and then contraction and formation of memory. The first pediatric patient who was treated ended up becoming very famous. Her name is Emily Whitehead, and she has a website and a blog and a foundation that raises money for cancer research, and she is now about 10 years cancer-free. What was also remarkable about CAR T therapy is that it really achieved a combination of what targeted therapy can do in terms of the frequency of initial responses, but also achieved what biologic or immunotherapy could do, which is sustained remission. And so if we compared the latest chemotherapy and biologic therapies at the time for pediatric relapse refractory ALL, we see that at the time, clofarabine and etoposide in combination with Cytoxan yielded a median overall survival rate of 2.5 months in a similar disease population. Blinatumomab, which is a bispecific antibody directed to CD19 that forms a bridge to T cells with CD3, led to an median overall survival of 7.5 months, and a CAR T cell therapy treatment single infusion led to overall survival at 6 months of 90% and median overall survival of 19.1 months. So this was really remarkable and led to the FDA approval of CAR T first in pediatric patients with relapsed refractory ALL. Now, what about the mechanisms underlying how CAR Ts work? I showed you that they had very different PK and PD than what a typical small molecule does. And it turns out that this is actually important for how they function. So David Porter and Carl June and our group at Penn first showed that this long-term persistence of T-cell in CLL patients was associated with durable remissions. So if you look at just the PKs of these cells, so the initial expansion and how long they stay around as memory, and you group them by the complete responders versus the partial responders and the non-responders, you could see very clearly that the complete responders have this rapid initial expansion and then persistence of the cells. And so that became sort of part of the lingo around how CAR T cells really work. And it's true, particularly for BB zeta CARs, but I would say that it's not entirely true for all CAR T cells that have different signaling domains. So it turns out that this 4-1BB CAR really has to have this marathon effect and has to stay on for a year or 6 months to a year in order to get that complete response. In contrast, CAR T cells that have a co-stimulatory domain derived from CD28, in this case, KTEX19 or Brexucell in adults with ALL, patients also needed to have this rapid expansion of the CAR T cells, but none of the 28 patients in this recently published study had measurable CAR T cells at month 6 or 12. And so although the expansion is initially required, that persistence seems to depend on how the CAR T cell affects the actual biology of the T cells, how long they need to persist, and what their anti-tumor dynamics are. This slide also shows that rapid expansion is associated with the main toxicities of CAR T cells, which are cytokine release syndrome from rapid immune activation, as well as neurologic events. Now, axi-cel, again, CD28 bearing CAR, even though there's not this persistence for years the way there is with BB zeta, that anti-tumor efficacy is actually maintained at 2 years. So you see this this sort of target Kaplan-Meier curves here, where you see that the duration of the response kind of flattens, median overall survival also flattens. And so if patients achieve and stay in a complete response at 15 months or so, they have a very good chance of staying in that complete response. So I've mentioned to you a couple of times that this difference between CD28 and 4-1BB seems to actually affect the biology of the T cells. One of the first projects I did when I arrived at Mass General, and I got my first very talented graduate student, actually did an in vitro comparison and used technologies that hadn't been available to us before then. And that was using both bulk RNA sequencing and single cell RNA sequencing to do a very rigorous comparison of how CAR T cells bearing a CD28 domain differed from those containing a 4-1BB domain versus those that didn't have co-stimulation. And so she made all of these CAR constructs and transduced them into T cells using the same process and the same lentiviral vectors, and then stimulated them with antigen. And the first thing that she saw in principal component analysis is that really stimulating through a CAR is very different than stimulating through the T cell receptor. In this case, it's using anti-CD3 antibodies, so it's not really an MHC plus peptide combination, but it is still signaling through the T cell receptor. That's the first principal component that came out. And then the second one that came out was really how long the duration of stimulation was. Was it 4 hours versus 24 hours. And so then looking at these T cell activation scores, she was able to show that really all of the CARs generated a similar T cell activation score, and this is probably driven by the CD3 zeta molecule in each of the CARs, whereas the T cell receptor stimulation really yields a much higher sort of potency activation of the T cells in terms of how many signaling pathways it activates and how much gene expression changes after stimulation to the T cell receptor. The other thing that we found is that 4-1BB CARs seem to behave differently than the rest. So if we look at single cell RNA-seq in a Tisney plot after stimulation of CARs that either bear 28 zeta, BB zeta, or just the zeta domains, we see that the 4-1BB CARs tend to cluster together. And then if we looked at unsupervised sort of topic analysis, it looked like 4-1BB specific CARs had a particular gene signature that we then also were able to recapitulate using artificial antigen presenting cells that had 4-1BB ligand transduced. So it seems that 4-1BB stimulation in particular seems to polarize T cells to become more Th1-like and actually suppresses Th2-like behavior. And so we see less production of Th2 cytokines like BB zeta in CD19 CARs after stimulation, in CD19 BB zeta CARs after stimulation with CD19. And this is true no matter what the antigen specificity is, and it was also true no matter what the affinity of the antigen specificity is. And so it really seems to be dependent on the co-stimulatory domain that is embedded into the CAR. And so just some thoughts about the data that I've shown you so far is that this expansion of the CAR T is probably required across classes to have anti-tumor efficacy, and that the intracellular domain of the CAR has a role in determining the persistence of the CAR T cells, its memory formation, and overall its fate. However, there are also additional genetic background in the transduced T cells that can also influence efficacy and perhaps toxicity. And some of the things that we've been looking at recently are the effects of clonal hematopoiesis, germline alleles that may be associated with differential function and memory formation of the T cells, and then gene edits or sort of accidents of nature where lentiviral vectors have, say, knocked out a certain gene in the CAR T cells and resulted in sort of uniquely super responders or unique toxicities. So based on where we are now, here's what I think of where the challenges are for CAR T cells in liquid tumors versus solid tumors. And I'm going to tell you first about our work in liquid tumors, which takes on a slightly different flavor than what our work is on solid tumors, which is what I'll close with. So in liquid tumors, we have a high potency biologic drug. That high potency is also associated with significant toxicities. This is what makes CAR T cells sort of not widely available in the community yet, is the management of the post-infusion cytokine release syndrome and neurotoxicities. Now, it's possible that this will get better with standard medical management and consensus guidelines and so on. But at the end of the day, it can be a little bit challenging to develop more CAR T cells for liquid tumors without having a deep understanding of how to manage the toxicities. The other issue that has emerged is that in some patients, they may have an initial response, but then their tumor relapses and it no longer expresses the target. And so that's known as antigen escape. And again, based on lots of prior work in infectious disease and HIV and tuberculosis and many others, we know, and even in cancer with small molecule drug inhibitors, we know that targeting a single pathway can result in sort of selection for pre-existing mutations or compensatory pathways that evade that selective pressure. And so, of course, naturally for CAR T cells, one way to engineer this is to start to have dual or multi-targeted CAR T cells targeting multiple antigens at the same time. And that is some of the work that's ongoing by us and others. Now, in lymphoma in particular, we are also observing that not all patients will have an initial response. So somewhere between 20 and 30% of patients don't respond at all. And there are multiple hypotheses as to why this happens. And we've been using the term lately of T cell fitness, which is sort of the quality of the incoming T cells. And this could be related to prior exposure to chemotherapy or simply the presence of a hematologic malignancy that exerts suppressive pressure on the T cell compartment. There's also been the question that's been discussed a lot, particularly by Crystal Makel, of T cell exhaustion, that engineering T cells with CARs and having continuous constitutive expression of the antigen and continuous exposure to the antigen can result in T cell exhaustion. And so ways of embedding rest into the CAR T cells, either using drugs or regulatable receptors, is a potential mechanism to enhance T cell memory and persistence and overcome exhaustion. And then finally, actually CARs, because they're synthetic molecules, they can be prone to immunologic rejection by the host, particularly if the host has not had lymphodepleting chemotherapy beforehand. This is a particular issue, of course, also for allogeneic products. And so making CARs to be more humanized and less immunogenic may also be a way of increasing their persistence in more patients. But the fundamental challenge for all of this is actually just identification and validation of more tumor-specific targets, because in the end, we could overcome antigen escape and we can use medical management to target, to manage potential toxicities, and also use small molecule drugs and engineering strategies to overcome T cell fitness issues, exhaustion, and rejection. And so I just wanted to highlight a couple of the studies that we're doing, first in the clinical setting and then more in the laboratory setting. So my colleague, Matt Fragault, who is my former fellow and is now a star in his own right, did some of the first clinical trials of TisaCel in secondary CNS lymphoma, and now has an ongoing trial in primary CNS lymphoma that is now finished accruing, and he'll be presenting at ASH in Atlanta this year. And what was remarkable here is that CAR T cells were actually quite effective at crossing the blood-brain barrier and treating disease even in the CNS. And so even though small molecule drugs may not be as effective as crossing this blood-brain barrier, CAR T cells can be quite effective in this scenario. And there's actually not any increased neurologic toxicity. It seems to be more related to the systemic disease burden, the antigen burden, and that seems to be driving more the T cell expansion and the toxicities rather than the location of the tumor. And so that's been very encouraging. And we are hoping to be able to, right now, CAR T cells carry a limitation of use where they're not allowed to be used in primary CNS lymphoma. And so we're hoping that this can become a modality that is of potential use for patients with CNS disease. One way that we have that we are exploring in terms of managing toxicity, and this is just one of many, there are several other investigators who are looking at other pathways, including GM-CSF and IL-6. One pathway that we decided to pursue was disruption of the IL-1 pathway using anakinra, which is the IL-1 RA, which competitively inhibits IL-1 beta signaling. And we think that giving anakinra prophylactically, meaning initiating it at the time of T cell infusion and continuing it for 7 to 14 days may actually prevent the onset of cytokine release syndrome and neurologic toxicity. It's too early yet to talk about data, but this will be presented at ASH in Atlanta in December. So let me tell you a little bit about some of the new targets that we've been developing for liquid tumors. We identified CD37 as a potential therapeutic target in the context of CAR T cells. CD37 is a tetraspanin family protein, so same family as CD20, but it's involved in cell membrane organization and signaling and modulating cell adhesion, motility, and proliferation. Like CD20, it's expressed in B cells during most of their lineage, but not in pre-B cells, so not of clinical use in ALL that we would expect, and not in plasma cells either. And what was interesting also about CD37 is that it seemed to be aberrantly expressed in several different patients that were tested in the literature with T cell lymphomas. And so this gave us a potential way of treating patients or enrolling patients who had not only B cell malignancies, but also potentially T cell and even NK T cell lymphomas. And so we first decided to look for CD37 expression in a variety of patient-derived xenograft models that were provided by David Weinstock, and also primary tumor samples of patients with CLL, and by immunohistochemistry in patients with peripheral T-cell lymphoma. And we identified that CD37 expression was very common in mantle cell, pretty widely expressed, almost 100% expression in patients with CLL. And we don't have high numbers yet, but we are planning on putting that data together, that it was expressed with some frequency of 20 to 40% in patients with peripheral T-cell lymphomas. And so based on this, we opened a phase 1 basket study, which I'll tell you about in a few minutes. We also showed that CAR37 was effective in xenograft models of both mantle cell lymphoma and patient-derived xenograft models of mantle cell lymphoma. And so we see here that comparing CAR37 to CAR19, these CARs are basically equally effective in cell line-derived models, and also equally effective, and if not a little bit more so, for CAR37 in patient-derived xenograft models in mantle cell lymphoma. And then looking at some models of T-cell lymphoma, in this case, CD37 positive prolymphocytic leukemia, also provided by David Wanstock, we were able to show that CAR37 was able to induce remissions in these mice where CAR19 had no effect because TPLL does not express CD19. Now, at the end of this experiment, actually, many of these mice ended up relapsing, and it looks like they relapsed with CD37 positive disease. We think that's probably because CAR37 does not engraft for as long in mice as it could potentially do in humans, because mice are not natural hosts for human T cells. Based on the xenograft data and data that's already been published that I will not belabor, we submitted our first IND targeting all hematologic malignancies expressing CD37 with our CAR37 construct. And so this is a 600-page IND that talks about the CMC process of how we will manufacture the CAR T cells. That's being done by Jerry Ritz and Sarah Nikiforo at DFHCC. And then our clinical collaborators, Matt Fergalt is the clinical PI, and we are also co-investigators with our bone marrow transplant team, Eben Chen and Arij Aljawari, as well as the rest of the PMT group at MGH. And so that protocol is open to enrollment. There have been 5 patients treated, and that data will also be presented at ASH in Atlanta. Next, I wanted to tell you a little bit about our efforts in multiple myeloma. So multiple myeloma has been a disease it's a plasma cell disorder, as you know, plasma cell malignancy. And recently there have been CAR T cells targeting BCMA that have been approved and that induce very rapid and frequent remissions in patients with relapsed or refractory multiple myeloma. Now, the current FDA approved CAR, which I showed at the beginning, is based on a murine anti-human BCMA single chain variable fragment fused to a BB zeta signaling CAR. Now, the issues with BCMA CARs is that they don't seem to be curative. So the median progression-free survival is about 11 months with these CARs. And it looks like patients relapse mostly because they lose CAR T persistence and not because they lose BCMA, although a small portion of the patients relapse with BCMA negative multiple myeloma. And so the approach we took before we knew what the mechanisms of relapse were going to be were that we decided to look at molecules that were related to BCMA. And these were all TNF receptor family members. And the 2 main closest homologs were BAFF receptor and TACI. Now, BAFF receptor is expressed mostly in immature B cells, but TACI has a similar expression profile to BCMA. And they both BCMA and TACI provide survival signals to plasma cells. And what was exciting to me about targeting BCMA and TACI is that we could use their natural ligand, which is called APRIL, which is made by myeloid cells in the bone marrow. And they share this natural ligand. And so we could build a CAR based on a shared natural ligand and target both BCMA and TACI while also keeping all human sequences so that there was no murine single chain or camelid-derived single domain antibody. And so we initially tried to use a single molecule of APRIL fused to our BB zeta backbone CAR construct. But that was actually not enough to induce anti-tumor effects in xenograft models. And so then what we did was we used the trimeric structure of APRIL and made a trimeric APRIL CAR, which we called TriPRIL for short. And so this CAR now targets both BCMA and TACI. And I'll show you just some of the murine data for this, which has also been published. So making this sort of concatemerized truncated APRIL molecule fused to a BB zeta CAR, we were able to show that in murine xenograft models where plasma cells expressed BCMA, that BCMA CARs were very effective. This is the data showing you that monomeric APRIL CAR was not enough to induce responses in these mice, but that our trimeric APRIL CAR actually was. And then we took it a step further to say, what happens if BCMA is lost, which we engineered using CRISPR-Cas9 editing of the multiple myeloma cell line. And then we treated these mice with BCMA CAR. Again, it's no longer effective because it doesn't have a BCMA, but our trimeric APRIL CAR was able to induce responses in most of these mice. And so based on this, and of course, additional data, which I'm not going to go through in great detail here, we were able to open a phase 1 study of tripro CAR T cells in patients with relapsed or refractory multiple myeloma who've had at least 3 prior regimens of systemic therapy, including a proteasome inhibitor, an immunomodulatory drug, and anti-CD38 antibody. Importantly, prior anti-BCMA therapy is allowed. I'm not showing you a whole landscape of all the treatments for multiple myeloma, but there are several, including antibodies and antibody drug conjugates that target BCMA. And so because our CAR is able to also target TACI, we figured that it was both ethical and interesting and potentially of clinical benefit to include patients who've had prior anti-BCMA therapy. And this trial is now open to enrollment. So what about solid tumors? Because I kind of ignored the right side of the slide the first time you saw it. In solid tumors, we have a similar issue in that the main challenge is identifying tumor-specific targets. But in addition to that challenge, what we've seen kind of across the field in CAR T cells for solid tumors is a fundamental lack of bioactivity. And the truth is that we still don't know exactly what's driving that, although people have sort of converged on the same consensus hypotheses. Some of it could be that we're not picking the right targets, that we don't have the right co-stimulation, that T cells don't traffic very well into solid tumors, or that they're becoming exhausted in the tumor microenvironment. The other issues with solid tumors are that there is antigen heterogeneity, and so even if you identify one tumor-specific target, it is likely not expressed throughout the tumor, and so then you have to identify more than one tumor-specific target, which becomes an additional challenge. And of course, what everybody thinks is the major issue, which I agree with, but I think there are others, and this is potentially targetable, is that there is a highly immunosuppressive tumor microenvironment that really impairs clinical benefit from CAR T cells, even if there is a direct anti-tumor killing effect. So we decided to start in glioblastoma, probably one of the most difficult solid tumors to treat, partly because of the blood-brain barrier, partly because it has a highly immunosuppressive tumor microenvironment, but it is an area with a highly unmet clinical need. It's almost universally fatal. It's the most common primary malignant brain tumor. Median overall survival is still 15 months, which hasn't really changed very much in the last 20 years. And we are also not the only investigators who are targeting glioblastoma. And there are multiple targets that are being looked at in glioblastoma, which makes it amenable to multiple different approaches. The other thing is that because it is a localized tumor, it becomes amenable potentially to local regional infusions of CAR T, and you could still get a clinical benefit as opposed to widely metastatic disease where you can't necessarily control trafficking and where the CAR T cells go. And so this is a list of potential antigens that are being targeted. And I feel like we should also just make note that glioblastoma is so far the only disease, solid tumor disease, where CAR T has actually induced a complete response. This was done at City of Hope by Christine Brown and Behnam Beatty, where they gave a patient multiple intratumoral and then intraventricular injections of a CAR targeting the IL-13 receptor alpha-2, and the patient had multifocal disease, both hemispheres and spinal cord, and actually had a complete response for about 7 months, indicating proof of concept that CAR T cells can in fact target a solid tumor. It just may require the right antigen, multiple doses, and potentially local regional injection. This patient ultimately relapsed with IL-13 receptor alpha-2 negative disease, again indicating that targeting multiple antigens is going to be necessary in all likelihood to be able to get sustained anti-tumor effect. So the antigen that we went after is a bit controversial. It's EGFR variant 3. And the reasons it's controversial is because it is not expressed homogeneously, and its expression can vary even within a glioblastoma during the course of treatment. But the reasons EGFRB3 were attractive to us is because it's an oncogenic mutation, so no normal tissue expression because it itself is an oncogene, and it's an oncogene that results from a mutation in the extracellular domain that is immunogenic. So there's a deletion of exons 2 through 7, and there's insertion of a novel glycine residue, and so this makes this is amenable to making an antibody and making a very specific chimeric antigen receptor that will only target the mutation. Because it's an oncogenic mutation, again, no expectations of on-target off-tumor reactivity because it's not expressed in normal tissues. But the challenge with it is that is expressed only in about 30% of glioblastoma cases, and its expression is heterogeneous and can vary over time. So our initial work back in 2012 to 2015 was published in 2015, involved rational development and characterization of a humanized anti-EGFRvIII CAR. So we started with an antibody that had been generated in Pittsburgh. It was called 3C10. We generated single chain variable fragments, and then in collaboration with investigators at Novartis, we humanized it to be called 2173. NIH had also made one called 139, and we tested various different signaling domains in intracranial models. Now, in these intracranial models, we used CAR-Ts plus tumor plus temozolomide, which is standard chemotherapy for glioblastoma. And in fact, none of these CARs were curative without the temozolomide in these models. But with temozolomide, the CARs with the 4-1BB zeta signaling domain were actually curative quite early on. And so that's the backbone that we chose to pursue. The next thing that we were really concerned about was making sure that we really only targeted EGFRvIII very specifically and not EGFR wild type, which is expressed in many, many normal epithelial tissues, including the entire gut, the lungs, and the skin. And so trying to get the most separation of binding of the 3C10 from wild type EGFRvIII to wild type EGFR to EGFRvIII became the top priority. And so as we humanized the single chain variable fragment, we also wanted to maximize this differential targeting of the mutant versus the wild type EGFR. So this actually ended up turning down the affinity of the EGFRvIII specific CAR. And so then we needed to retest its specificity in mouse models. Retest its specificity in mouse models. So here we used intracranial MRI of mice and showed that injecting mice with IV untransduced T cells had essentially no impact on glioblastoma growth, whereas injecting them with CAR EGFRvIII was able to induce responses as measured by MRI in these intracranial mouse models. We also developed a new skin toxicity model where we grafted human skin onto the backs of NSG mice, let those mice recover, and then injected CAR T cells, either specific for wild type EGFR or EGFRvIII CAR. We were able to show that this was an adequate model because wild type EGFR resulted in toxic rejection of the skin graft with keratinocyte apoptosis, which is basically GVHD of the skin measured histologically, and there was a rapid infiltrate of the CAR-Ts into the skin graft. The time point on the left is at 14 days, the one on the right is at 2 days after injection. Whereas the EGFRvIII CAR looked clean in that there was no lymphocytic infiltrate of consequence in the skin graft here, and that the epidermal dermal junction stayed intact, and there was no rejection of the skin graft and no keratinocyte apoptosis. So based on these data, we opened a phase 1 study. This completed accrual, enrolled 10 patients at Penn. Of note, it's such an unmet need that we had more than 400 patients send us their tumor samples asking if they could take part in the study. Approximately 20% of those had EGFRvIII expression. We enrolled 10 patients, and then we gave them a single injection of CAR T cells targeting EGFRvIII into the peripheral vein. This was published in 2017. The lead PI was Dana Farber. And Dana Farber is a neurosurgeon. One of the wonderful things about working with him was that he performed tumor resections or tumor biopsies in many of the patients, and 7 of the 10 after having received the CAR T cells targeting EGFRvIII for various clinical reasons. And so that gave us an opportunity to look at the tumor and assess whether EGFRvIII expression had changed after CAR targeting, whether it was specific or if it was also wild type EGFR amplification that changed, both by next generation sequencing and by immunohistochemistry. And it also gave us a chance to look at the tumor and its microenvironment histologically. So the first thing we found is that almost all of the patients had decreases in their level of expression of EGFRvIII, and this was specific to the mutated antigen because there was no change in EGFR amplification or wild type EGFR expression by immunohistochemistry. A couple of the patients had a relatively short window between their pre and post infusion resections. And so we were able to see in these patients is that the CAR Ts actually did robustly traffic into the brain tumor, or T cells effectively trafficked in. And there was a significant T cell infiltrate in many of these patients. But unfortunately, most of them were not CARs. They were, in fact, regulatory T cells, CD25 positive, FOXP3 positive cells that had trafficked in. And by looking at TCRV beta sequencing, we were also able to determine that most of these regulatory T cells had come from the circulation. And so they were not present in the apheresis bag, but they were somehow recruited after the infusion of the CAR T cells. The tumor itself also underwent a change in the immunosuppressive microenvironment in that it upregulated PD-L1. And this was seen across the patients, but just I'm showing you this one example where it was most dramatic and where there was the closest time between the pre and post resections. And so when I came to MGH, I wanted to ask the question is, could we design a CAR T cell that would target a second antigen, hopefully all that wild type EGFR that you saw there, while also either bypassing or even better co-opting those infiltrating regulatory T cells. And so the strategy that we came up with is to build on this EGFR B3 CAR, but then also have a second transgene in the same vector that would secrete a T cell engaging antibody molecule. We called it a BiTE before we knew that it was trademarked by Amgen. So I apologize for that. It's in the paper as a BiTE, but now we call it a TEAM. And so this TEAM molecule can basically target the undruggable wild type EGFR because it gets produced locally by the CAR T cell. It is produced in very, very small quantities. I'll show you how we tried to quantify it. And because it's so small, it gets rapidly cleared from systemic circulation. So the idea would be to have the CAR T cells injected locally where they secrete the T molecules and target both EGFR V3 and wild type EGFR. And if any gets stuck in systemic circulation, it won't be stuck for very long because it'll be rapidly cleared by the kidneys. The other exciting thing about T molecules is that they can also induce Tregs to actually become killers. Tregs actually express perforin and granzyme, and in the presence of these T molecules, they do actually kill, not in a serial fashion, the way you see with cytotoxic CD8 T cells, but they do get at least one round of kill in that. And so that gave us hope that we could potentially turn all these infiltrating Tregs into at least transient killer cells. And so these were the constructs. They start to get pretty big when you have both an EGFR V3 CAR and a secreting bispecific T cell engager or T molecule. And we had a control CAR where we had the T molecule become just CD19 specific, so we could control for the size. And then we had just simple CARs, second generation CARs targeting each of the molecules in turn. And so the first things that we verified is that this the T molecule actually, that both ends of it worked. And so here we're looking for binding of the of the T molecule. Every time I move the mouse, I move the slide. The T molecule could bind the antigen portion that we were interested in. So K562 cells transduced with either CD19 or EGFR. You could see that by flowing soluble T molecule over these antigen expressing targets and then staining for the histidine tag, we could find the teams binding to those target cells. And then looking at the other business end is, could this T molecule bind to T cells? The answer was also yes, we could get binding to T cells, although we had to concentrate the supernatant, and that's because the CD3 binding is relatively low affinity. We also did some calculations over time using T cells to measure how much BiTE could be secreted or TEAM could be secreted over time. And we came up with somewhere around 83 picograms per mil per hour. Per T cell per hour were being secreted in this construct. And so that gave us sort of the initial flavor of what the overall log of exposure is going to be for humans who get CARs that secrete T molecules. We were also able to show by confocal microscopy that we could get sort of teams that bound both to untransduced T cells. And so we get soluble secretion of the team, and then they coat an untransduced T cell. And here the green is staining for the T molecule with biotinylated EGFR, and the red is the M-cherry of the construct, the lentiviral vector construct that stains all the transduced cells. And so you could see that the T molecules form these little yellow islands. So some of it gets basically secreted into an autocrine fashion where it comes back and stains the T cell that made it, but enough of it gets produced in a paracrine fashion such that untransduced bystander T cells are also coated with T molecules. We were also able to use transwell experiments where CAR T cells were on the upper chamber and then tumors were on the lower chamber, combined with just untransduced resting T cells. And so the bites that were produced by the CARs actually crossed the transwell and were able to bind the untransduced T cells and then mediate anti-tumor activity. And we could see that with both brain tumor cells coated on the tissue culture plate, as well as U87 cells that were, these are brain tumor cells that were transduced to express CD19. And so here's the EGFR bite, and the purple is the CD19 bite. And we could see that both of them could initiate tumor killing if the tumor had the target. And I want to also say that all of this work was led by Brian Choi, who was a neurosurgery resident in my lab, who is now an assistant professor at MGH. And Brian was also able to leverage some of his connections in neurosurgery to get some patient-derived xenograft models, including with Haruki Wakamoto, who gave us the BT74 line. And so here with these neurospheres, we were also able to show that although CAR T cells targeting EGFRB3 had minimal impact, although there is heterogeneous expression of EGFRB3, CAR T cells that secreted the EGFR-directed T molecule were able to significantly shrink this neurosphere over time. And again, most glioblastomas express wild-type EGFR greater than 90%. And so really being able to target both of these antigens was key. Now, what was interesting is that actually just having the bite alone was not enough. So having this T molecule with untransduced T cells actually leads to sort of an exhausted phenotype, whereas having the CAR secrete the bite leads to much more of a central memory phenotype of these cells. And you could also see that by having the CAR secrete the T, we were able to reduce exhaustion based on phenotypic markers. So less PD-1 expression, less TIM3 expression, less LAG3 expression, compared to just exposure of T cells to T molecules. And so based on these data, we think that there's actually use in just having CAR T cells secrete a T molecule, regardless of the antigen specificity of the CAR. And this was proven true in some of our xenograft models, which I'll go through here towards the end. So the first thing was that CAR teams were able to induce responses in mixed tumor models where the same cell line, some of it expressed EGFRB3, so the CAR target, and some of it did not express the CAR target, but all of it expressed wild-type EGFR. And what we were able to show in these intracranial models where CAR T cells were giving into the ventricles of the mice, again, this is a benefit of having a neurosurgery resident in the lab, they do intracranial injections and they learn how to do that in mice, is that these CARs that secrete the bite EGFR were able to induce responses in all of the mice. And in fact, what was even more interesting is that in a glioblastoma cell line that does not express any wild-type EGFRB3, we were still able to induce responses in about 80% of the mice just by virtue of the secretion of the bite or T molecule targeting EGFR, indicating that the co-stimulatory domains of the CAR, coupled with the antigen specificity of the T that's secreted by the CAR, is enough to induce anti-tumor responses. Now, what about toxicity? I mentioned that we were very hopeful that T molecules would be rapidly cleared from circulation and not cause on-target toxicity, but that's just a hope. We wanted to show that experimentally. And so what we did is we leveraged or brought back our skin graft tumor model, where we engrafted human skin onto the back of NSG mice, and then gave CAR T cells intravenously to sort of maximize exposure of the skin graft to the CAR T cells. And again, we had CAR T cells targeting wild-type EGFR, or CARB3 secreting T molecules to CD19 or to EGFR wild type. And what we saw is, again, this is our positive control CAR targeting EGFR, where we see rapid T cell infiltration and rejection of that skin graft with keratinocyte apoptosis. Our EGFRB3 CAR we already knew was safe, did not cause any on-target toxicities in humans, but we showed that in the same mouse model. And then our CAR that secreted the EGFR bite molecule or T molecule also was safe in this model, where there was no keratinocyte apoptosis, T cell infiltration, or rejection of the skin graft. And so based on these data, we have now an IND in review with FDA to be able to open this study, a phase 1 study in patients with EGFRB3 positive and even EGFR V3 negative, just EGFR positive glioblastoma. And so I've told you about several of our translational ambitions. We have several new targets that are in various stages of the translational pipeline. We have been treating patients with CD37. We've just opened multiple myeloma. We have a dual specific CD79B, CD19 CAR for which we are intending to file an IND by the end of this year. Our glioblastoma study is in review. And then we're also leveraging the engineering aspects of CAR T-cell therapies to make enhanced safety and degradable CARs using drug-inducible CARs and also gene-edited CARs in the course of the next couple of years. This is a very rapidly growing field, and this is really just to give you a sense of how much this field is growing. If you plot CAR T-cell searches in PubMed in the going back to 2007, you can see that we're in log phase growth. What's interesting is that there's about an equal number of clinical trials listed of CAR T cells on clinicaltrials.gov, also in log phase growth. So I think the next 5 to 10 years will tell us a lot about how amenable this therapy is going to be to use in multiple different indications. So I'll just conclude with a couple of thoughts that CAR Ts targeting CD19 have really, and now BCMA, have entered mainstream clinical practice in academic medical centers in the US and also starting in Europe and Australia and Japan. There are many ways to engineer next generation CAR Ts for different tumors to have different functionalities to overcome resistance or to prevent relapse. Manufacturing and financial toxicities are ongoing issues. This is still an expensive proposition. And there are a lot of small steps with manufacturing that can make a very big difference in terms of clinical impact. There's been rapid growth in our field, and it's really converging new biologic advances with rapid translation and impressive bioactivity. And that is for both toxicities and responses. So it's a very active field. And with that, I'd just like to thank you for your attention and also thank my lab members, my collaborators, and my funders who are all listed here. Thank you very much.
Thank you, Marcella. That was truly amazing. You've really focused on the important issues, and I'm really blown away by everything that you've been able to accomplish. We don't have a lot of time. I'm going to start with the first question from someone named Tian, who's asked how much antigen expression is needed for a CAR T cell to recognize and kill the target.
Yeah,
and what about the affinity? How does that affect the activity?
Yeah, so we don't know an exact number, and it may vary depending on the CAR and the tumor cell, actually. So we've done some experiments with CD19, for example, where it looks like even antigen that's not detectable by flow cytometry or immunohistochemistry is detectable by CARs. And so we've estimated that it's about 100 molecules of CD19 to be able to get killing by a CD19 CAR. But it's not clear that that number is going to be the same for all CARs and all antigens. But I think it does give us kind of a window that it's somewhere in between a T cell receptor, which only needs 1 to 10, and immunohistochemistry or flow cytometry, which needs more than 1,000. So I think it's going to be somewhere in the middle.
Great, thank you. And Diane Krause asks, says, great talk. CD37 is not completely specific for B cells. Are there concerns about CD37 and anemia because it's expressed on erythroid progenitors and or effect on T cell function?
Yeah, that's a great question. So we did a lot of preclinical work looking for potential fratricide of CAR T cells against other T cells and against hematopoietic progenitors, against isolated immune subsets, including neutrophils, monocytes, macrophages, and K cells in peripheral blood. And we actually didn't see anything specific. But based on what we're seeing now, which we'll talk about at ASH, I don't think that those in vitro assays are necessarily sufficient because you get such a rapid expansion in the patients that those issues start to pop up a little bit more.
Okay, Nolan Beatty is asking about the CD37 relapse, and he's wondering if it was because of cells in the TME like MDSCs or tumor-associated macrophages?
I think I might have missed something because I don't think that we've talked about CD37 relapse.
Yeah, because you haven't, you don't have the data.
Yeah.
Okay, I'm not sure what tumor he was talking about. So let's move on. Rebecca Boyarsky asked, how do you assess potential off-target effects? For example, GTEC shows CD37 expression in brain and spinal cord, but it doesn't seem to be an issue in the clinic. Early in the process of identifying CAR T-cell targets, how do you decide which ones to look at?
Yeah, that's a great question. It's so hard. There's no straight answer for this, and every sponsor does sort of what they can and what they pursue. I mean, definitely the availability of large data sets that describe expression and the Human Protein Atlas data sets. That's sort of where we start. Sometimes though, the tissues are not defined by cell type. So for example, like intestine, for looking at most B cell targets, like intestines will come up, and that's because there's so many B cell and immune cells when you look at like a whole piece of gut. But we don't actually think that they're intestinal epithelial cells. And so then we do sort of targeted in vitro assays looking at primary cells derived from the tissues that we're concerned about. So there's a combination of sort of mining data sets and our own pursuit of primary in vitro assays. There are also now some contract research organizations that will provide either primary cells or G-Rex, for example, where they express sort of every peptide in the human genome, and then you can flow your CAR targets for that or assess for potential cross-reactivity. I wouldn't say that there is one standard, but there are several possibilities for looking. Yeah.
Great. So we have time for one last question, and it's from Jun Ren, who asks, why did you need TMZ in GBM for the CAR T cells to be effective? And I'll ask at the same time, have you thought about combining the CAR T cells with something that directly makes the tumor more visible to natural, other natural recognition, maybe?
Yeah. So, usually for the patients, they've already had temozolomide, and so we went after patients, or we included patients in terms of enrollment eligibility who had already progressed after temozolomide. And so then there's not as much clinical benefit to them continuing the temozolomide because they have toxicity. In the mouse studies, the tumors just grow, that particular tumor line just grew too fast. And so temozolomide was sort of just enough to slow it down to have the CAR Ts be able to have their effect. And Judy, in terms of adding in other drugs, I think that would definitely be our goal. I know that Don and Carla have continued now with EGFRV3 CAR plus checkpoint blockade. But I don't really know how it's gone. My sense is that it hasn't been transformative because I would have heard about it already. And it's not clear that checkpoint blockade has single agent activity in glioblastoma, which is the problem, is that there's not a lot of agents with single agent activity in these tumors. And so you could potentially combine it just to increase the potency of the T cell, but then you also have to take into account sort of what toxicities are you adding, and will that confound your measurements of toxicity related to your CAR? So I think there are trial designs that could begin to ask those questions, where we have first single agent CAR to prove safety, and then a combination with a clinically used drug to try to improve potency. So I think those are very reasonable things to try.
Great. So for the people whose questions weren't asked, Claudia will send your questions to Marcella to address. And I just want to thank you for an awe-inspiring talk, and wish you all the best with what you're doing, and I expect to hear great things soon.
Thank you so much.
Okay. Bye, everyone. Have a good afternoon.